Hello. In this series of videos, I'm going to show you how you're going to proceed with the annotation exercise. There will be different bioinformatics tools that you're going to use for each part. Let me show you something that I already should be familiar to you now, and hopefully to make you feel better about time when you don't have a lot of time to do something that is tedious, or you don't want to devote a lot of time to something that is tedious. So I'm going to share with you a slide you've seen before from this class about using BLAST. And this is when I told you about the 6-frame translation. Keep scrolling down. Slide 26. There you go. So remember this? I told you, oh, imagine you were given this DNA sequence. What are the 6 possible proteins it can make? And you're like, oh my God, I'm not going to have time to do that. Yeah, you won't. You're not going to do this by hand. This part is to show to you how BLAST comes up with 6 possible proteins that will come out of one string of DNA that you enter. So I explained to you the 3 different frames, reading frames. So because we don't know what are the 3 letters that make a codon here. Is it these 3? Are they being read together? Or is it these next 3 that are being read together, or the next 3? And if you keep going, if you start on the 4th letter here, you're going to end up as frame 1 again. So when you look at this string here, this DNA sequence, if you start on the first base, you go CTC. If this DNA sequence was the coding strand, then that means that the mRNA is the same sequence as that, except that the T's are U's in mRNA. So let's consider this the coding strand. And so the mRNA will be CUC. And you translate it here, C. That's first letter. Here's the second letter, and then third letter. So CUC encodes a leucine, the amino acid leucine. And then you go to the second triplet if you're still going to keep reading, because see, you would form this string here. So that L is for the leucine. The second codon would be still on that reading frame, ACA. And then you keep going, reading each 3, comparing it to this table. This table is called the genetic code, and it's pretty much universal, except there are some exceptions. But we're going to use the universal genetic code. So some organisms use this differently, and I'm only telling you this out of curiosity. They are exceptions. Okay, then what if this sequence here is read in this triplet here that's marked in red? So I don't want you to feel like when I give you a DNA sequence, which I will, you have to do this procedure by hand on reading each 3, comparing each codon to the genetic code. I will show you a tool that will do this for you. Isn't that handy? So what you would want to search is translate DNA. That way it does it for you. Conveniently enough, I have exactly the one that I wanted to show, but there are other DNA translation tools. So now I'm curious about this one. But here, let's go to this one, focus. So I click on that, and in here it tells me enter DNA or RNA sequence. So let's, I want to grab the sequence that I have. Well, let's put something else there. It feels like typing random stuff. And here it's saying the output. Do I want it to be the full word, or the compact is the 1-letter code for amino acids? So amino acids, they have their full name. their three-letter name and their single-letter name. And do I want it to say stop? Do I want it to have a hyphen? Let's see. And do I want it to also show the nucleotide sequence? I'm just going to show you the default. So right now, all I did was I entered the DNA sequence in here. And here's the part that I told you that the genetic code, there's a standard one, that's the one we're using. And if you're curious, like, what else is out there? Then you drop down the menu. You're like, oh, mitochondrial DNA reads that table differently. But if you want, you can play with that. I'm just going to do using the standard genetic code. So here is the result. Look at that. Oh my goodness, that's so good. That did this so fast. So these results here are this sequence read in each of those 6 frames. So the computer read these 3. So read those 3, and then it skipped the first one and read the second 3. Maybe let me do it again with the nucleotide sequence so it's easier for you to see. Look at that. So it read those 3, and down here, this is the sequence that would come out of translating this one starting from the first nucleotide. And then this hyphen here, don't be confused by that. In this case, so I'm not aligning sequences, right? This is not an indel. That alignment is saying that there would be a stop here. So UGA is a stop when there's an mRNA that this would say UGA, that's a stop codon. And if you're like, oh, I don't remember what that hyphen is for, okay, then don't despair. Just click instead of compact, where it would put a hyphen instead of saying stop. You can go to verbose, translate, and then it will say, oh, it's a stop codon. So the protein would stop, the translation would stop there. So maybe I'll keep on this part because I felt it was useful. So on frame 2, you might think, oh, is it reading 4 letters? No, what it's doing is ignoring the first letter, and then the first 3 is TAG, which UAG is also a stop codon, and now it's going to translate down. And when it says open reading frames are marked in red, then it has a reason to believe that this part would be the part that would encode the protein. This will become clear once I run something with a longer sequence. You will see. What is? You can actually click on it. All right, that's some new feature. I was curious what it does, but I still don't know what that means. Oh, it's saying that the reading frame would start here. So that would be my protein. Because over here is a stop codon, so it thinks that it would start here, except that proteins start with methionine. I don't even know why it marked as a possible protein start.